So yeah, so let's get going. So we're going to start off with Randy. I'm really excited about these two talks today. Randy, who's an old friend, a long way back, and professor of psychiatry at Mass General, and she's also the director of imaging science for the McCance Center, and has a bunch of other titles. But most important thing is she's running our imaging efforts as indicators for brain health at the McCance Center, and we.
You. Here is slides. Okay. So it's hard to talk and see your faces. So please unmute yourself and feel free to ask questions or interrupt at any point in time. I like that better than just talking. So please feel free. This work that I'm going to share with you about, some of you might have heard me speaking on this topic last year around this time. And this is the work that I do when I'm not wearing my McCance hat. This is the work that I do wearing a hat that's in psychiatry and radiology, together with a collaborating team at Martinos. This program project work is funded by the National Center for Complementary and Integrative Health. And I like being part of this team, partly because everybody is awesome to work with, but most impactful for what might be interesting to folks here today is that this work was really right at the tip of the sword of where advances on the technical side of imaging is pushing us to be able to know and understand things about how brains function. In this particular study, we're looking at we're studying a hypothesis about the coupling between a top-down intervention, in this case, stress reduction therapy, mindfulness-based meditation, and how it interacts with a specifically targeted bottom-up or body-based intervention, in this case, transcutaneous vagal nerve stimulation, or abbreviated TVNS for here, and seeing how that might help us to study and understand mechanisms underlying the disorder of migraine, but also how they might be impacting the expression of the disorder by using those treatments and studying people before and after those treatments. And so while not as severe of an impact as dementia or stroke, migraine headache is a really prevalent worldwide disorder that causes significant morbidity and mortality. Not mortality, but morbidity. It really has a negative impact on people's ability to work, play, and just given the number of people who are even here today, at least someone on this call likely has migraines themselves or has a friend or family member who does. And so it's very relevant in the world. Let me see, I have no disclosures. This is work, like I said, done by a fabulous, fabulous team. And unique to this program project, there's a single experiment, which I'll be going over in detail in a moment. And then the various projects, which have distinct leaders, focus on attributes of which of the data that's acquired they are focused on creating the detailed experimental design for, and then analyzing the data, presenting it. But we work together as a very cohesive team. You probably know a lot of these folks. And we include some of our most wonderful team at Martinos, who are very good at helping us to use the most state-of-the-art image acquisition and image analysis techniques. Many of you may not have met Dr. Zev Shulman-Olivier, who's our director for the mindfulness-based meditation. If you are interested in that kind of treatment for anything in your life, you should definitely look up that facility. They do wonderful work, and he's been a great collaborator throughout. And our team of postdocs and research assistants and research coordinators is just superb. I'll be highlighting the work of all three of the postdocs have put effort into giving me some slides to share with you today, even though it's still pretty early days. So how do we think about our work? It starts out with a vision that that this, as I said, this top-down and bottom-up approaches to treating migraine may be synergistic in how they might impact the parts of the brain that are different in people with migraine compared to people who are healthy. And in specific, hypothesizing that certain regions of the brain, including the thalamus, the hypothalamus, the posterior insula, are areas where there are neuroinflammatory processes that might be going on, that these are brain regions that are thought to have a phenomenon known as central sensitization. That is to say, they respond abnormally to a range of normal inputs. It's also pretty well documented that the autonomic nervous system is dysfunctional in people who have migraine in more subtle ways, but in measurable and consistent ways, that being mediated through some of the brainstem nuclei. And so when we do our work, the idea is that we bring in treatment with mindfulness-based meditation, that is a top-down or mediated by our more cortically centered brain through the medial prefrontal cortex, the anterior cingulate, the anterior insula, and the impact of that brain activity on these more deeper brain structures and specifically down to the brain stem may have a moderating or mitigating influence. That's our top-down pathways. And then conversely, the ravens, the respiratory gated auricular nerve stimulation, has its input to the brain through a branch of the vagus nerve. This is like a little tickle of current. The gizmo that goes in your ear is a lot like your earbuds if you have iPod headphones, any kind of headphones. And the R in terms of respiratory gating has to do with the finding from our team that if you provide that gentle stimulation during the exhalation as opposed to inhalation, it seems to have a more robust effect. And so the idea is that by stimulating the autonomic nervous system through that branch of the vagus nerve, that then stimulates moving up through the brainstem, the noradrenergic and serotonergic neurons in the locus coeruleus and the raphe, respectively, and that ascends through those same brain regions that we're trying to target with the mindfulness-based meditation, that they interact between each other's, and that's what we're trying to study, is can we see that with the imaging? So we have a big study, about 120 people being recruited and randomized into one of 4 groups. For the group that we are studying the mindfulness-based meditation, the top-down, they get a sham type of stimulation in their ear. For the ones that we're studying the ravens, TDNS, they get a kind of a sham meditation and education control that we use that has no breath work or no meditation to it. We use both those 2 controls as in a third group, which are non-specific effects, and then look for a synergy between the 2 active arms in the group that gets them both. So this is a very colorful flow diagram of what happens through this long, many month study. Importantly, not shown on the slide is we do have an additional cohort of 30 people that we're going to take through this, just the first top row that gets screened and tested through these baseline visits that are healthy controls that are aged and sex and comorbidity matched to the cohort of migraineurs that we'll be recruiting. And the screening visits include a testing for taking blood for genotyping. One of the genes, the TSPO gene, is known to affect the binding affinity for the PET ligand that we use. And so if a person doesn't have either of the alleles that makes them a high or a moderate, might have high or moderate binding affinity for that ligand, then we don't take them through the PET visit because we know we won't have a signal there. But everybody goes through rigorous screening, and importantly, they're doing ongoing daily diaries so that we have a very accurate count of the frequency of their migraine headaches throughout this whole time period. Once they're screened into the study, There's 3 different visits where we collect the main imaging and behavioral data. A PET MR with a brain PET that also has fMRI and spectroscopy and other kinds of scans collected in between the PET acquisitions. And then the fMRI visit is in our ultra-high field 7 Tesla scanner for doing the brainstem mapping. The behavioral visit is an autonomic nervous system assessment that gives us detailed information about how their autonomic nervous system works. Once they get all of those visits completed, they go through a very carefully choreographed 8-week treatments, 8 weeks of treatment that includes individualized training sessions, group sessions, as well as booster visits with a lot of care and attention from our clinical team. And you can see the little dollars and which parts are in person and which are online. Thanks to COVID, our team has had to do a lot of pivoting and adapting to be able to continue the study through the pandemic, which they've done an amazingly good job of. I just want to say thank you, team. Once they finish all the treatments, they return for an identical set of data gathering visits, behavioral visit, the PET MR, and the 7 Tesla scan. And then we continue to stay in touch with them for 3 and 6 month follow-up visits. I do not have data on those things or the outcomes of treatment. I'm going to be focusing in today's talk about some of the data we've gathered during the PET MR visit and during the 7T visit. So just a moment of background for those who may not know about using this PET ligand, the PBR28 ligand. It's used for looking at patterns of neuroinflammation. Importantly, it's a highly sensitive ligand, so that if you have a healthy brain and you get scanned after this ligand is injected, there will pretty much be no binding, has very low basal expression in a healthy brain. But it has been studied in a number of different disorders where there are very powerful reasons to believe that there are lots of lines of evidence to indicate that there's been neuroinflammation, specifically targeting microglia in many cases, but it may not be exclusive to that. So positive findings with this ligand have been found in experimental autoimmune encephalomyelitis, in multiple sclerosis, HIV, encephalitis, and even in Alzheimer's disease. There has been some preliminary work in animal models of pain, but now in the hands of Marco Loggia and his team, also in a couple of chronic pain disorders. And so that was the tool that we brought to bear here. In this first preliminary analysis that Ludovica pulled together for us, we followed the same methods that are previously published, so that all the details of methods are well documented, and we didn't do anything different from them. In this case, where the results I'm going to show you are from a comparison of 19 patients with migraine disorder compared to 13 healthy controls. Migraines are more frequent in women than in men, as you can see from our distribution of the patients, the participants. Importantly, we have people who are relatively earlier in the disease, people closer to the age of onset of in their 20s, all the way through their 50s, and a balance in terms of their injected dose. Replicating this earlier finding, what we can see is that the migraineurs have more binding of this ligand in a number of brain regions. It's significant even in this early, partial cohort as we're collecting the data. And when Ludovica, this is the result from the whole brain scan, whole brain analysis. When she did a more careful parcellated the data and looked into those clusters only in regions of interest, so those areas that I was showing you in our hypotheses. And that's what's depicted here. On this graph, the binding of the signal is shown on the y-axis, and then each of these letters depicts one of the different brain regions: the amygdala, the nucleus accumbens, subcallosal cortex, but you can see in the gray markers are the healthy controls, and in the red, the migraine patients, that there are differences already that are hopefully going to be fully statistically significant by the time we get our full cohort, but it's looking very much like in each of those regions, we are seeing significant increases in the patients compared to the controls. And that was pretty much what we were hypothesizing. And it depends, if this continues to be true with the full cohort, it demonstrates, this is in vivo evidence, that migraine is accompanied by prolonged signs of neuroinflammation in humans. And that gives rise to opportunities for strategizing how to moderate that inflammation as a potential therapeutic target. So thank you, Ludovica, for that good work. While they are in the scanner for the PET scans and that data is being collected, we have opportunities between the PET acquisitions to run the fMRI scans. And so together our team designed the newest adaptation of a task paradigm for studying interoceptive attention. And so interoception is the process by which the nervous system senses and integrates signals originating from within the body. So in prior publications using interoceptive attention to the heartbeat, we've seen nice activations in the bilateral insular cortex. So in our task, we wanted to make maybe even a more robust task. So we had blocks of time where we asked them to attend to their heart rate, and other blocks of time we asked them to attend to their breath, their breathing. And we compared those blocks to times when we asked them to pay attention to the sounds of the scanner, or blocks of time where we asked them to pay attention to just the little textured background screen that was up on, so visual stimulus. And we used verbal prompts to cue them what they should be paying attention to, and then had rest between that. And literally the situation in the scanner is unchanging throughout this entire acquisition. So the sounds are always the same, the screen is always the same, and then the only thing that changes are those cues, the word cue that tells them what they should pay attention to, or just a blank if they're resting. And so when you do the fMRI statistical analyses and you plot the activation, where are the signals higher during the times when they're cued to pay attention to their heart and their lungs, compared to the times they're cued to pay attention to sight or sound, we do see the expected activation in the bilateral insula. This is a preliminary data from 18 of the migraine patients. This is, again, early results, which mostly are telling us that our paradigm did not break anything and that we are seeing the expected activations, but more information will come when we have the full cohort and compare this to patients versus controls. But even within these 18 migraineurs, we did see a relationship in terms of that activation to the number of migraine scans, the migraine headaches that they had experienced in the days prior to the scan, suggesting that there is some kind of relationship between the experience of the migraines and the circuitry that it helps them to attend to their interoceptive sensations. It's going to be really interesting to study this data after the treatment. This is all, like I said, the baseline. So if you haven't had any of the mindfulness-based meditation, none of these patients have had, as participants have used the TVNS. And so this is the signal that we'll be looking for, those shifts. And give a shout out to Michael for his work putting that together. The last of the data I want to share today is collected in the 7 Tesla scanner. This is work that Sarasa on our team has done. She's actually, I think I skipped a slide. Wait a minute. There we go. So while they're in the 7 Tesla scanner, we do a number of different kinds of acquisitions. This data is designed to help us to map these very tiny, discrete pathways. In this case, we're looking at activating the pathways responding to an innocuous and non-painful electrical stimulus applied to the subject's foreheads. And so this is just an on-off design, turning the low electrical stimulation on, off, a little tickle of current, and then looking in the brainstem nuclei that would be relaying that signal into the brain. And in the first of the data that Sarasa has processed, she's been able to find a signal. It's still the work that goes into the signal processing is very intense because this part of the brain is tough to image. It moves and pulsates with breaths and heartbeats. And so you have to do corrections for the physiology data that's being collected simultaneously with the fMRI data. But it's showing in what will be, I hope, a landmark finding that we can see that activation, and then hopefully be able to study how similar or different it is between the patients and controls and how it might be adapted after treatment. Finally, Sarasa uses data collected in that same time that's used with a diffusion sequence to look at measures of integrity of the white matter bundles. And specifically in this particular analysis, she was looking at, in this little blue box, you can see where my cursor is pointing, these tiny little threads. Those are actually the trigeminal nerve roots that are thought to have carry those central sensitized signals in patients with migraine. This picture is from the T1 weighted MP rages. These are the lower two images are derived from the diffusion scans, the FA map, and then a color-coded FA map that tells you about the directionality of the fibers. This two tiny little red dots, those are the bits of the trigeminal nerve that she's then using her skills to analyze diffusion data to say, what are the characteristics of the nerve fibers that are running through the trigeminal nerve root right there, and is there any relationship of the parameters extracted from the diffusion data that map to their clinical characteristics. And what she's been able to find already is a significant negative correlation between one of those metrics, the axial diffusivity, which is a metric of integrity, with disease duration, so how long the patients have had their migraine disorder, and also with the number of headache days that they experience per month, saying that there may be something about the disease itself and the characteristics of that nerve root. There's also beginnings to be a signal in this cohort. This is from 16 patients. We don't get the diffusion in every single patient. It's one of the last scans collected in the block. So if for any reason they can't make it through the whole sequence, a set of sequences, this is one of the ones we don't always get. So there's a slightly smaller N here, but showing that the time since the last headache also seems to have an impact on this metric. And so it's very exciting to be able to get signals that are meaningful from with such terrific spatial and time resolution. And so we are continuing this work, and there's a lot left to do in this study. We still need lots more patients, so lots more participants. So if you know too much to recruit yourself, but if there is somebody in your network or in your family or it's within reasonable distance and you think that they would enjoy participating, please do send them our way. And with that, I will pause and see if there are questions.
Thanks, Randy. That was great. Amazing how many angles are coming in, in terms of mindfulness to devices, to imaging, to different disorders. And I'm wondering, I'll take the first question. And if anyone has questions, please put them in chat, or you can use the raise hand symbol. And Christian, right after my question, I'll have you speak. There are different ways to stimulate the vagus, by massage, by breathing techniques, as you know. And I'm just wondering, how does this TVN device compare to just, the old-fashioned way of just belly breathing, 6 breaths per second? Is the massaging, the ventral vagus? How do these things compare? Have you looked at that?
I think whatever works. So I'm a big proponent of doing what you can, but But Vitali, if he had his disclosure slides up, he'd show that he is an advisor to this company, Cala, who's marketing this device for broader use. And so we are actually doing some research with them in addition. But I guess what I would say is gizmos help people be more more consistent with something.
Yeah.
This device is actually quite comfortable. It is, when you use it and you're meditating, it's kind of like, it's very pleasant. It's not an unpleasant experience at all. It's actually very, it in some ways it just feels nice. So I don't know. I don't know that anybody's put them head to head to say, how is this work versus how is that work in the same trial. What I can say is that when these devices have been used in small clinical trials, they have been effective. The precursor devices. One of the co-PI on my project, Ronald Garcia, he has actually developed a device like this at home in his home country in Colombia, and we have used that device in some of the early stage work. I know it's also Jill Goldstein and her team are using it for studies in depression. There's, I think, a couple of different groups ongoing who are finding this device to be useful. So
yeah, it's PTSD, anxiety, depression, epilepsy. I mean, vagus nerves everywhere, right? So that's great. John, go ahead.
I was going to say, I'm happy to talk with anybody about the device and
cool.
also the what we're doing with it and why we think that it's actually advantageous. And we will be doing head-to-head, comparisons of these types. We're just not there yet. We're still developing dose, frequency, all sorts of things that may be different across different disorders and also by sex. So we are in the weeds on developing this device for depression. Vitali does it for migraine. We're also looking at comorbidity with hypertension, really interested in that. And Rudy, we're also very interested in tackling memory issues because of the shared circuitry with stress. So
that's great to know. So I'll be in touch about that, and others feel free to reach out to Randy or Jill about this device. It'd be great. So one last question from Christian.
Hi, very nice study. I had a question about your microstructural data. I mean, that looks really interesting. And I know to get this kind of changes on an anatomical level, there already has to be quite a lot of change in the brain. We had done a similar study where we did mice exercise and tried to measure microstructure. And we were kind of trying to figure out what is changing in the brain on a cellular level. Can you speculate on that? Do you think there's more myelination? Do you think it's the way the fibers are orientating, or what do you think is on an anatomical level happening that you see in this improvement or in the pathology?
So I would be speculating wildly because I don't have, there is no real data that would say it's this or that. I can say that the being able to measure these things at all is already like right at the envelope. And because we can't, like in the animals, you can actually do the imaging and then look at the histology, look at the microstructure, be able to make the hypotheses. And so we rely on, the collaborative studies with colleagues. We're doing this kind of work in animal models to study that aspect more specifically. I will say that there are multiple diffusion metrics that can be looked at. And I, for one, am still trying to have my colleagues who are the deepest experts in that domain help me to understand why this one and not that one in this case or that case. I have not gotten a hugely convincing answer, which is why I'm saying preliminary. I think probably the most meaningful thing that will come out of this study is if and how if we can show that there's a difference between the patients and the healthy controls, and then anything that's different actually shows a normalization, moves more like the controls after treatment. That's the signal we're looking for, because even though we can't explain it, it helps to know that there's a spot there that's doing something that actually makes a difference, because that helps us to target and then partner with the people who can do the invasive studies to look for those more mechanistic results. It's also, we're beyond the sensitivity of the PET data when we look at that trigeminal fiber. Our PET data isn't that, doesn't have that level of resolution.
Yeah.
But there may be a signal in the brainstem that corresponds to what's happening in the trigeminal system that we might be able to see with that microglial activation with the PET, which is why these studies are being done in the same patient population, going through this comparable study. So I hope when I'm talking to you guys in 5 years, when we have all the data, I can answer your question really well.
Thank you for a great talk, Randy. I think your talk stimulated my vagus nerve, so that's good. So I feel better already.